Hello and welcome to ACUR at ANU, to this student's presentation. We would first like to acknowledge and celebrate the first Australians on whose traditional lands we meet and whose airways we share. We'd like to pay respect to elders past and present. My name is Jules Lumbers and I'll be chairing this session today. Firstly, I'd like to introduce our first presenter, Emma Sinn. Thanks, Emma, when you're ready.
Hi everyone, let me share my screen. Okay, cool. Can you guys see that? Okay.
Yeah.
Okay, so hi everyone, my name is Emma and I'm a current honours student at the Heart Research Institute based at the University of Sydney. My honours project is focused on understanding the role of normal ageing in the development of a subset of heart failure known as stiff heart failure. Firstly, what is heart failure? Heart failure is a disease in which the heart can't meet the metabolic demands of the body. Patients commonly present to hospital with symptoms including lung congestion and shortness of breath, particularly at night. This disease disproportionately affects older people over 65 years old, and it's actually the leading cause of hospitalizations for people in this age group, both globally and in Australia. In 2019, 8 Australian deaths per day were actually due to this disease. So it's a very severe disease and it's definitely going to affect our futures. Well, hopefully not our futures, but Australians in general. There's 2 types of heart failures and they're delineated by ejection fraction. The ejection fraction is the amount of blood that's pumped out of the left ventricle, and that is oxygenated and it goes to circulate around your body. On the left, you can see heart failure with reduced ejection fraction, and that describes systolic dysfunction or the inability to pump. Since there's many treatment options available for this type of heart failure, it's actually decreasing in prevalence, which is really good. However, there's no treatment options available for the other type of heart failure with preserved ejection fraction. So it's actually now the predominant form of heart failure, and this is the heart failure that I'm focusing on. It's also called stiff heart failure because it describes impairment in the heart's ability to relax whilst filling is still healthy. So since it can't fill properly, it still cannot eject enough blood to provide for the rest of the body. Heart failure with preserved ejection fraction, or HFpEF, is a cardiometabolic syndrome. It affects mostly older people with comorbidities including obesity and diabetes and hypertension. Aging is a significant factor in disease progression, and model systems thus far have only studied HFpEF induced in young mice and not isolated the effect of aging on diastolic dysfunction. Given Australia's aging population, I believe that focusing on aging is the key to maximising our futures. So we ask, what is the role of aging? In the heart needs to consume a lot of energy, and this is in the form of ATP. It needs a lot of energy to fuel the continuous pumping. Most of the ATP is generated by a process called oxidative phosphorylation, which occurs in the mitochondria of the cardiomyocytes in the heart. Nicotinamide adenine dinucleotide, or NAD, is a key cofactor for energy generation. However, studies have identified that during both aging and in HFpEF, NAD levels are severely depleted. That suggests that NAD depletion may cause decreased ATP generation, which underpins cardiac stiffening and subclinical diastolic dysfunction that we see in the elderly. And this is a process called cardiac aging. So this brings me to my aims and hypothesis. We hypothesize that replenishing myocardial NAD using the precursor nicotinamide riboside, or NR, for 6 weeks will reverse cardiac aging in mice and improve markers of basal metabolic health and diastolic dysfunction. We aim to answer this by assessing basal blood glucose and insulin and cardiac function through echocardiography, or I'll refer to this as echo. To answer these aims, we compared 3 groups of mice. We compared healthy 18 to 24-month-old mice, which is the equivalent of 60 to 70 human years, with or without NR treatment, with healthy 15-week-old mice, which is the equivalent of young adult 20 to 30 years. Firstly, to assess basal blood glucose and insulin, the mice were fasted overnight and a tail snip to collect blood, and the blood was measured using a clinical glucometer. The level of insulin in the blood was measured using an anti-insulin ELISA plate, which measures the concentration of insulin in the whole blood. We found that aging didn't induce a significant effect on the blood glucose or insulin, and this was expected because the older mice, whilst they were older, they were not diabetic. However, we did identify that cardiac hypertrophy or enlargement of the heart can be caused by aging. The heart weight and tibial length ratio is widely used to assess hypertrophy of the heart, and our graph here shows that the older mice had significantly increased hypertrophic heart compared to the young mice. However, when we compared the NR treated versus non-treated old mice, there was no difference. So that indicates to us that NR does not actually affect the hypertrophy of the heart due to age. Next, onto the echo parameters. Echo is a cardiac ultrasound imaging technique, so we can see the function of the heart, and the mice are sedated during this, but they're still alive. On the left, we can see the image of a young heart, and the red brackets show the wall thickness. On the right, the same, but the red arrows, you can see even visually, the thickness is a lot more in the old mice. The graphs show the summary data that overall, there is a significant increase in wall thickness, both during contraction or systole and relaxation or diastole for the old mice. When we normalize this back to the tibial length, we can see that the old mice have significantly increased hypertrophic walls, which supports the findings we found before with the heart rate and tibial length. And that's important because it suggests cardiac thickening, which is related to fibrosis and diastolic impairment that's caused by age. Once again, we compared the NR in the NR treated old mice versus the non-treated old mice, and we saw that there was no effect on the wall thickness. Another important measure of cardiac function is the IVRT, or isovolumic relaxation time, and that is the time that it takes for the heart to relax at the end of systole contraction and the start of diastole. On the left, we can see I've circled the IVRT looks visually a lot shorter than on the right in the old mice, and in the graph we can see that the old mice had a significantly longer IVRT time. That indicates prolonged relaxation, which is a marker for diastolic dysfunction. When we compared the NR treated and non-treated old mice, once again, NR did not have a significant effect on the IVRT. One of the most important echo parameters for assessing diastolic function is the global longitudinal strain, or GLS. This is a measure of presystolic function, or the functional capacity for the heart to deform or change in thickness throughout the cardiac cycle. On the left, which is the young mice, you can see the green lines are tracing the thickness of the wall, and you can see that the walls are changing in thickness quite a lot throughout the cycle. On the right, you can see the thickness is not changing as much throughout the cycle, and that's expected with aging. When we have a graph which shows the overall difference in GLS between the young and old groups, so GLS is expressed as a negative number, and a more negative number indicates better cardiac function. So we can see that the old mice had significantly worse cardiac function, significantly more diastolic impairment compared to the young mice. When we compared the aged mice with and without NR treatment, there was once again no significant difference in the pre and post conditions. However, we can see that there seems to be an upward trend in the GLS for the control mice, which means there's a trend for the GLS to get worse in the old mice, whereas it seems to be getting better in the aged mice, but once again, it was non-significant. So we do need to do further stats to determine if it's realistic. These are two other echo parameters that showed no difference in age, so I'm not going to explain them fully. And overall, my studies have found that aging causes increased cardiac stiffening and thickening, i.e. diastolic dysfunction, or a trend towards stiff heart failure in mice. This is particularly relevant with the conference theme, as our population is aging, and diastolic dysfunction is a common manifestation of cardiac aging. And without intervention, our older population might have an increasing risk of stiff heart failure. However, my study did not find any evidence that NR affected these parameters or diastolic impairment due to age. This is because maybe we needed a longer treatment time or other things such as how echo can be very operator dependent. Some future directions, some studies have suggested echo parameters don't always correlate with myocardial fibrosis, so it would be useful to do fibrosis staining and metabolomics to look at cardiac energetics and the NAD metabolism itself inside the heart. So that's all. I'd like to thank my supervisors and my lab group members. Thank you, and any questions?
Thank you very much. Okay, so we now have 5 minutes for questions. So first of all, we'll pass to judges. I know that Dr. Lee Gao has just joined us, so thank you for joining us. Maybe Yash, did you have any questions at this point? Oh, sorry, Yash.
No,
sorry.
not yet. Yeah, probably I joined a little bit late, but yeah, thank you.
That's okay. You can you'll be able to view the video, the recording after, so that'll be fine. Sorry, Chamakara, did you have any questions?
Yeah, that's a great presentation, Emma. I really love the pitch and energy in you. So I got like 2 questions. One sort of the more technical question. So you already mentioned that you didn't get any significant difference in GLS, I guess. So you mentioned that you don't have enough sample number with that. That's maybe the reason, one of the reasons that you haven't got any significance. Do you know any other reasons? Like even if you increase the sample number, still it would get significant. So do you know any other reason that it wouldn't be significant? Like many other factors that affects this?
I don't know that much about stats, so I'm not that sure, but GLS...
No, I mean, like not statistically, I'm asking like, you know, many other factors what would affect this other than the sample number. Mm-hmm. Like not statistical, like, you know, more biological.
I'll just go back to the image.
Yeah.
So as you can see, to analyze the GLS from the ultrasound, you like basically draw a line around the walls of the heart. This is kind of a fiddly process, and sometimes there's like a shadow if, for example, the mouse had like a mole or like a hair on their chest. So I think that that could also affect how well we can analyze the GLS. So I think that might be it. But also, I think I mentioned before that perhaps the NR treatment might not have been for a long enough time. We treated them for 6 weeks, but which is kind of long in mouse time, but it might be different if we did more like several months, because that would have a bigger effect overall. And also when we compare with the metabolomics of the heart to see if the NAD levels actually changed with NR, then that would give a better indication of the actual effects.
It's perfect. And one simple question, what are the other future directions that you look on this research? What sort of?
So I mentioned the histology that I would like to do because we really would like to see if there is visible fibrosis in the older mice and not just, I don't know, more muscle or something that's causing the increased thickness. So we'd like to stain for fibrosis, and we'd also like to do metabolomics looking at the NAD consumption, and we'd also like to do proteomics to look at NAD consumers. So studies have found that some NAD consumers really increase in aging. So there's a molecule called CD38 and it's expressed on inflammatory cells, and it actually consumes like 100 molecules of NAD for each like one enzyme or something like that. So we'd really like to see if the levels of that are increasing a lot, and maybe that's why NAD levels are changing.
Yep, that's perfect. Thank you so much.
Thanks for your questions.
Okay, we still have time for questions. So if anyone else had any questions, I can open it up. So if any attendees, any of the other presenters had any questions, you're welcome to pop them into the Q&A box or raise your hand and ask away. Otherwise, I will ask you, Emma, why did you choose this as your topic?
It was interesting. I did a summer project with the, sort of with my supervisors, and it was looking at heart failure the type of heart failure that this lab looks at, which is stiff heart failure. And then we were also considering the other factors that cause heart failure to be so severe in some people. And one of the main factors was aging, and the other one was like diabetes, hypertension. So I thought that the aging aspect was pretty interesting, considering aging is something that people can't really avoid with lifestyle factors. Whereas I guess diabetes can sort of be managed with other lifestyle factors or other treatment options.
Great, thank you. All right, it doesn't look like there's any other questions. So we might leave it there. Thank you so much. That was fantastic. All right, so with that, we'll move on to our second presenter. So Yash Malhotra, you are up next. Thank you when you're ready.
Thank you. Yeah, I will just share my screen. That starts on my last slide. Hi, my name is Yash. I'm a student from the University of California, San Diego, and today I'll be going over my thesis topic, which is reprogramming macrophages to resist atherosclerosis using GIV. Atherosclerosis cardiovascular disease remains one of the leading causes of vascular diseases worldwide, with approximately 18 million individuals succumbing to it yearly. It is characterized by the deposition of cholesterol plaques in the inner walls of the arteries, which results in the obstruction of proper blood flow. Macrophages are key cells in our innate immune system and play an important role in the development of atherosclerosis. In hyperlipidemic conditions, they scavenge harmful oxidized low-density lipoproteins, or OXLDLs, that accumulate in the aortic wall and transform into sticky foam cells. These foam cells continue to build up due to the uptake of cholesterol. And the lysis of these foam cells leads to a progression of inflammatory processes that ultimately results in the formation of atherosclerotic plaques. These plaques have a tendency to rupture, leading to thrombosis, which can ultimately impede blood flow. In this research, we present a novel atherogenic signaling pathway in macrophages, which presents a possible avenue to manipulate the disease. The pathway is initiated by the multimodal G protein activator known as GIV. So trimeric G proteins were first identified in the late 1970s as signal transducers downstream of G protein coupled receptors, or GPCRs. Signaling from G proteins is canonically activated by their corresponding GPCR receptors that serve as guanine nucleotide exchange factors, or GEFs, which allosterically release GDP from G proteins and allow the binding of GTP to create an activated state for downstream signaling. Alternatively, G proteins can also be activated by a group of cytoplasmic proteins known as guanine nucleotide exchange modulators, or for the purpose of this presentation, GEMs. GEMs possess an intrinsic ability of modulating G proteins and were first discovered only 10 years ago. GIV is one of 5 members of the GEM family and activates signaling downstream a diverse class of receptors such as toll-like receptors and receptor tyrosine kinases. Previously, our research group has shown that GIV is a potent inhibitor of cyclic AMP. And since cyclic AMP is an anti-atherogenic second messenger, we wanted to study in detail the role of GIV in the progression of atherosclerosis. To do so, we created a conditional macrophage knockout mouse line and performed a sequence of experiments that I will be going over. So for the purpose of atherosclerosis, cyclic AMP is important in preventing the disease as it suppresses innate immune functions and has been shown to prevent the recruitment of monocytes to possible atherogenic sites, as well as the conversion of macrophages into sticky foam cells. To study if GIV influences cyclic AMP in a high cholesterol environment, we performed a cyclic AMP measurement assay on peritoneal macrophages isolated from mice. So from this assay, we basically found that GIV significantly reduces the levels of cyclic AMP in a high lipid environment. As can be seen from this graph, where the levels of cyclic AMP is represented by red in GIV knockout mice and blue in wild type mice, the levels in GIV knockout are significantly higher. And just for the ease of this presentation, this color scheme of red and blue will be kept the same. So we believe that in the presence of GIV, the reduced levels of cyclic AMP are unable to play their atherosclerotic role, possibly resulting in increased recruitment of monocytes and even increased foam cell formation. To test our hypothesis that a reduction in cyclic AMP levels will lead to higher foam cell formation in wild-type cells, and to understand the possible phenotypic effect of GIV in a high cholesterol environment, we developed a model for foam cell formation. So in this model, peritoneal macrophages were isolated from mice and treated with ox-LDLs for 24 hours. The cells were then stained with an oil or red lipid stain and imaged. So as can be seen from these comparative images, where cholesterol is stained red, the extent of foam cell formation for wild-type macrophages was significantly higher as compared to macrophages from GIV knockout mice. Wild-type macrophages not only underwent foam cell formation, but had visible lipid droplets in almost all regions of their cytoplasm. Upon quantification of this data, we found that wild-type mice underwent foam cell conversion almost 5 times more than GIV-depleted macrophages. We believe this study indicates a significant phenotypic difference in the progression of atherosclerosis through the development of foam cell formations in GIV knockout and wild-type mice. When it comes to foam cell formation, improper cholesterol efflux from macrophages is one of the key contributors. We therefore hypothesized that the genes involved in cholesterol efflux were being inhibited by GIV to increase foam cell formation. To understand this, we ran a qPCR on wild-type and GIV macrophages stimulated with ox-LDL for genes involved in cholesterol efflux. To represent the data, a heat map was generated relative to the expression in unstimulated wild-type macrophages. So from our qPCR analysis, we found that GIV significantly reduces the expression of various genes involved in cholesterol efflux. In this heat map, red color reflects high expression and blue color reflects low expression. And it can be seen that almost all the genes involved in cholesterol efflux show significantly higher expression in GIV knockout macrophages. Of these genes, ABCA1 and ABCG1 are the most important, as they together account for almost 70% of all cholesterol efflux. Gene 3 and 4 are also important, as together they form a nuclear receptor core-repressor complex with GPS2 as its essential subunit. This complex not only increases cholesterol efflux, but it also functions as an inflammation repression pathway. Finally, LXRs, which are represented by LXR A and B, have been demonstrated multiple times to play a very important role in cholesterol metabolism. When activated, they induce a series of genes to upregulate cholesterol efflux and transport. To ensure that this gene expression data correlated with actual cholesterol efflux data in macrophages, a cholesterol efflux assay was performed. The assay was designed to quantify the amount of cholesterol removed from cultured cells in the presence of a cholesterol acceptor. As a natural acceptor of cholesterol in human plasma is high density lipoprotein, or HDL, we decided to choose HDL for this assay. From this figure, we see that consistent with the gene expression data, GIV-depleted cells have a higher cholesterol efflux as compared to wild-type cells when treated with HDL and HDL plus lipopolysaccharide, which is a bacterial ligand and an activator of TLR4, one of the receptors activated by GIV. Thus, it can be said with a high degree of confidence that GIV inhibits cholesterol efflux in macrophages, thereby promoting lipid accumulation and foam cell formation. In atherosclerosis, the lysed foam cells ultimately lead to the formation of atherosclerotic plaques. The formation of these plaques in the walls of the aorta is the hallmark of the disease and is what ultimately leads to the obstruction of blood flow via thrombosis. Based on our findings so far, we wanted to observe how GIV influenced the formation of these life-threatening plaques. To do so, an in vivo model was developed. In this model, aortas were isolated from mice fed a normal chow diet and a fat-rich Western diet for 18 weeks. The aortas were then stained to study lipid formation in the aortic roots and the walls of the aorta, and then subsequently imaged. From our imaging analysis, it was observed that the presence of GIV significantly increased aortic plaque formation in mice that were fed a high-fat diet, while this effect was less prominent in mice fed a normal diet. Comparative analysis displayed in this graph revealed that the presence of GIV resulted in a 3-fold increase in lipid formation in the aortas of wild-type mice as compared to the aortas of GIV-depleted mice. This highlights that GIV plays a role in promoting atherosclerotic plaque formation, and this role is more significant or influential in a high lipid environment. G protein signaling pathway represents one of the largest classes of targets in the druggable human genome. However, it has not yet been manipulated to regulate the progression of atherosclerosis. We believe that this study is important as it demonstrates a fundamental role of a new cell signaling pathway involving a G protein in our immune cells and has the potential to usher new therapeutic targets in the future to modulate lipid metabolism. I think this is especially important with respect to the theme of the conference because in today's fast-paced world, more people have a high cholesterol diet than before. So I believe it's extremely important to possibly identify targets to avert this condition. Thank you so much for listening, and I would love to answer any questions that you might have.
Great, thank you. Right on time. Okay, excellent. So I'd first like to pass to our judges if they have any questions. We have 5 minutes of question time. Do either of our judges have questions? Chamkara, I can see you're taking yourself off mute.
Yep, Dr. Lee, do you want to go first?
Probably, this is probably a little bit outside my expertise area, but I'd like to probably ask, can you just mention a bit more about how to use this technology in practice or change or help people to get a better health?
So for us, we so far have not moved into completely modulating the effect of GIV in terms of a therapeutic target. Right now, the possibility we were considering on testing further is to create a conditional knockout mice line further that has a double knockout with ApoE, which is commonly used to study atherosclerosis in mice. And once we are able to understand that model, since it more likely represents a human model, we can more realistically try to identify how this pathway can be modulated. Right now, I would say based on what I've presented so far, the only possibility that I can think of would be to control the expression of GIV via conditional knockouts in certain lysosomal cells. But then again, I don't know if this can be safely executed currently because our model is not perfect. We are developing the double knockout model for the past few months now because that matches the human atherosclerotic condition much better, and we'll be able to come up with more realistic data for that can then be used further to develop possible therapeutic targets.
Thank you.
Yeah, that's a great presentation, Yashaswat. I'm really interested in that, and you presented really well as well. Bit more technical, just remember when you present, make all the technical words more comprehensible to others as well, right? But it's still a good presentation. And I just want to ask, so there's a true reason why we are looking on macrophages in atherosclerosis because it's the most abundant immune cell type, right? But have you looked on any GI effect on other monocytes or any other immune cells? Do you have any idea about that?
Yes, we currently I'm also working on a project to study the role of GIV in netosis, which is in neutrophils. So we're still trying to identify the role there, as well as the role of GIV in the general circadian rhythm genes. So those are two things that our lab is further investigating.
Yeah, that's perfect. And a small question: you already showed us a small heat map. So did you? Can you go back to that, please?
Of course.
So you draw this using qPCR data? Is that what you said?
Yes, I did qPCR data, and then
so
the
what
data
values
was.
Are these? So what values are these?
It was qPCR data that was made relative to the expression of the wild type value, and the data. The CT values.
Fold changes?
Yes, the CT fold changes were standardized to 18S expression.
Okay, I just want to know these are fold changes or not. Yeah, that's perfect.
Okay, yeah.
That's great.
Thank you.
Great. Okay. Did anyone else have any questions? Any of the other presenters? No. I feel like I should ask you the same question then, Yash. what drove you to research this topic?
I'd say primarily two things. The first one was my interest for immunology and immune cell interactions, and the second, my interest for cardiovascular diseases. And this seemed like a perfect combination for the two. So I decided to go with this project when I joined the lab.
Great. Thank you. All right. Well, it doesn't look like we have any other questions. So we might leave it there. Thank you very much. Excellent presentation. So we are now going to move to our final presenter today. And this one is by Harold Ju. This is not related to cardiovascular. So this will be a little bit different, but it's going to be very exciting nonetheless. So this will challenge us all, get our heads into a different space. So I'm looking forward to this. Thank you, Harold, when you're ready.
Yeah, I'm going to share my screen. Let me know if you can see my screen or you can hear me, but I'll get started. Greeting, I'm Qingqi Zhu. My research topic is including occlusion in human post-estimation benchmark, a photoheuristic synthesis approach. This project is supervised by Dr. Liang Zheng at ANU in my honors year, and this research is in the discipline of computer vision and deep learning. Let's begin with my research question. I would like to build a synthesis pipeline that could generate datasets for the task of human pulse estimation on the occlusion scenario. It is expected to contain 4 ground truths. Even on the occlusion, it ought to be photoheuristic. Besides, it needs to be controllable in hyperparameters. Here is an overview of contents. The first section is motivation. We shall take a look at 3 key concepts. Then comes a section, synthesis pipeline, where I'd briefly discuss my methodologies. Thirdly, we'd cast light onto the experiment designs. I'd also demonstrate some fundamental results. At last, I'd summarize the discoveries and introduce future work. As for motivation, there are mainly 3 aspects. It's universally acknowledged that current deep learning-induced computer vision highly depends on large-scale datasets, which is often scarce due to various reasons. But we've got a graphical engine nowadays. Modern graphical engines like Blender, Unity, and Unreal are astonishing in the capability of simulate the real world. However, that's more than just using them to create synthetic datasets. Rather, its relationship with computer vision is actually a two-way benefit. Synthesis benefits learning in facilitating data augmentation, model selection, etc., while learning benefits synthesis in realizing the objective of being realistic. And next concept, human pulse estimation, in short HPE, is defined as given an image or video, localize the human joints, such as elbows and others. It is fundamental for many human-centric tasks, such as 3D human reconstruction or motion capture. Lastly, there are different types of occlusion in real life, as shown in the bottom right picture, but most state-of-art architectures experience heavy degradation at them. So recently, there is increasing research interest into this topic. I've completed 2 synthesis pipelines, which are of increasing functionalities. Here's an overview of the first synthesis pipeline. It is built on a previous work of my supervisor. It is named as Person X, a synthesis pipeline for the task of pedestrian re-identification. The first step is to load 1 character from Person X package, and it comes with pre-configured textures, animations, etc. This character will be placed into a scene and get animated. Next, we purposely include diversity, such as position, background, image resolution, etc. Afterwards, our capturing procedure will save renderings of different cameras as well as the ground truth of key point positions, then this process repeats for another character. However, along with fine-tuning this pipeline, we gradually touch its upper limits. One major issue is that poses generated from animation has a sparse and peaked distribution. And on the other, high encapsulation of person X leads to the lack of flexibility. Therefore, we come up with the second synthesis pipeline based on two new technologies, SMPL and spin. SMPL is a 3D representation of humans that inputs shape parameters, beta, a less time vector, and post-parameter theta, a 24/3 matrix, then outputs a blended and a skinned person. Spin is a deep learning architecture of 3D human reconstruction. It takes an image with a person, fits SMPL model onto it to estimate the shape and pose parameters. Here's an overview of the second synthesis pipeline. Given a sample like this from the COCO benchmark, whose pose can be used, will be reused, plus a few other processings, the final rendering can then give us an image like this. And each pipeline has generated one synthetic data set. The first pipeline delivers pose X 0.7. The left of this page shows some samples from it. On the right is some of its statistics. There is in total 120 characters involved and 15,000 samples have been obtained. The second pipeline delivers pose X 1.0. Similarly on this page, the pictures demonstrate some pose X 1.0 samples and the corresponding COCO images where the pose is mapped from. The right table tabulates some statistics. It's worth mentioning that pose X 1.0 contains two partitions, where the only high-level difference is what type of occlusion is included. This also proves our pipeline being hyperparameter controllable. How well do the proposed synthesis pipeline work? We have designed experiments to validate the system and evaluate their photoheuristic level. As for system validation, we propose to evaluate from two aspects, homogeneity and eligibility. Homogeneity means the state-of-the-art models are able to converge on synthesized data sets and give comparable accuracy. Eligibility means the performance trend of SOTA models on synthetic data set should be similar to that of benchmarks. We choose COCO as benchmark and choose simple baseline and HRNet as our state-of-the-art models. The right table tabulates the baseline, the different average percentage performance given by different settings of state-of-the-art models, training and testing on COCO dataset. Here is the visualization of system validation of pose X 0.7, namely AP, AP.5 and AP.75. The purple line is performance of COCO, the baseline, and the pink line is the performance of same setting, but on pose X 0.7. It tells that all SOTA models converge and give a slightly higher AP. As for the trend, it is overall a similar gradual increase, same as a baseline, with a minor exception, which is highlighted in this red circle. So to conclude, pose X 0.7 and the first synthesis pipeline behind it is homogeneous and partially eligible. The same procedure is reported here for pose X 1.0, and this time we see an almost identical trend in AP and a slight varied trend in AP 0.75. Thus, we can conclude pose X 1.0 And the second synthesis pipeline behind it is both homogeneous and eligible. We aim to synthesize photoheuristic samples, but how to evaluate this photoheuristic level? We propose to evaluate from both machine and human perspective. Human perspective or learning perspective means knowledge transfer. In other words, for a SOTA model learned from a real benchmark, how well it can perform on synthetic datasets. Human perspective, in other words, human perspective is in other words visual effects, which is a subjective judgment. The baseline is a cross-domain process where we train different SOTA models on COCO and then test on MPI, which is another real-world benchmark in HPE. Other experiment settings are identical to the system validation. Here's the knowledge transfer results. The gray bars are baseline. Orange bars are transferring from COCO to POSIX 0.7, while the blue bars are COCO to POSIX 1.0.
Thank you, Harold. That was really informative. An excellent presentation. Okay, we now have 5 minutes of question time. So first pass to the judges. Did judges have any questions?
Okay, I will go first. Yeah, that's a great presentation, Jinsu. So my first question is, so you already mentioned that you have transfer your pipeline or the knowledge to real life benchmarks as well. Can you explain any examples, like what sort of real life examples, benchmarks that you're gonna use?
Sorry, which kind of benchmark?
Like real, how do you use this your pipeline or the technology or the system that you have developed for real life benchmarks? Can you give me some examples?
Yes. So I guess I'm gonna focus on the second synthesis pipeline. So which I just briefly talk about. So these benchmark datasets, they have diverse images of humans in different environments, and these humans are giving different poses. My benchmark is my synthesis pipeline is able to extract a pose from a real image and mapping this pose to a fake person or a 3D representation of a person and put it into a random environment. And the reason I'm doing this is because in real data sets that in the existing benchmarks, there is very limited samples or person under occlusion. And if there is occlusion, then we cannot get the ground truth of this person. So for example, if a person is partially occluded by some other objects, then we cannot identify where his joints are. But if I use my synthesis pipeline, then this person, then we can still get this person's joints because we are in graphical engines, and we are in kind of like a guard mode. We can see these positions.
Yep. So much.
Yeah, maybe my question to you. Yeah, firstly, Ju, this is a great presentation. So probably my question is similar to the last one. How do you see the practical use of your technology? Which area you can use, and what kind of the benefits we can get from use of this technology?
Yeah, sure. So it's briefly mentioned in my motivation part. So the current performance of state-of-the-art models on existing datasets, they are kind of like saturating, and they have severe degradation. So the performance drops a lot when on the occlusion samples. But when using my technology, so my synthesis pipeline, the data set can be extended to include lots and lots of samples of occluded scenarios, and the models will be able to learn from these samples, and when they are deployed in real life, they will perform better when they see occluded samples.
Great. Does anyone else have any questions? Excellent. All right. Well, we've only got a minute left, so we might leave it there. I'd like to thank all of the presenters. You've all done a wonderful job. Well done. Thank you for tuning in from all over the world, because our presenters are all over the place. And I'd like to say a very big thank you also to our judges, Dr. Li Gao, and also to Chamikara. Thank you so much for your time, volunteering your time here. I wish everyone the very best. Thank you to those who have tuned in and watched. And please enjoy the rest of the conference as well, because we are not yet done, and the award ceremony will be this afternoon as well. So please, do not just turn off now and leave us. We're not yet done. This has been such a wonderful, wonderful conference. So I've really enjoyed it so much. So thank you, everybody.
Thank you.
Thanks.
Thank you. Have a good day.
You
Thank
too.
you.
Thank you.